Okay, good afternoon. We're going to begin. Today's discussion concerns what, I don't know, to me, and I'm sure to many of you, is one of those incredibly exciting, I don't know, frontiers, I guess you'd call it, of science and medicine. It seems to me a field that is like sort of exploding, not only in the literature, but in concepts, technology, understanding, and application. And where it's exactly going to lead to seems to be challenging, uncertain, and yet incredibly exciting for the reasons which are going to be presented today. This, in case anybody hasn't been here before, is the Brooklyn Bridge, which sort of symbolizes the purpose of this course, demystifying medicine, trying to bring concepts of basic biology and medicine, medical problems together in a format that excites and stimulates your interest to learn more, not to sit and take copious notes. Can you put the next one up? Okay, so just by way of introduction, having in the beginning gotten very, excited about this brave new world, it's a brave new world that's kind of been rediscovered in concept and enormously enhanced by technology. But probably the first person to ever put his finger on this concept is the Russian Nobel laureate Ilya Mechnikov, who over 100 years ago, as they say, he was getting old and perhaps a little bit on the senile side, and he had this concept that the intestinal organisms were not just a cesspool, but they were really part of the entire living system and influenced health and influenced disease, of course. And he had the concept that a good, healthy life, which in large part, from his standpoint, had nutritional components connected with it, that would help to prolong life and to make life better. And being a microbiologist of great fame, he had discovered an lactic acid forming organism, Lactobacillus bulgarica, which he described as the bacillus of long life. And for most of his life, he drank sour milk, which contains the organism. And unfortunately, he died at the age of 70. But nevertheless, in those days, lifespan was sort of around 40 to 50. So maybe it did have some. At any rate, his concept and his personality and his incredible science, he's the father of one of the major branches of immunity. At any rate, his impact was tremendous. And for a long period of time, particularly in Europe and to a lesser extent in the United States, yogurt, a whole industry came up because of this. Kefir was manufactured, I think, in Japan, I'm not sure, and widely used, and people drank it all, sort of the idea. And oh, by the way, one of the side features of this course is a quotation by a Swiss nutritionist, Werner Koloth, who wrote, "Much is known, but unfortunately in other heads." Now, Koloth is the man who first used the term probiotic, which will emerge in today's discussion. And for those also interested in history, the microbiome as an entity, as a term, was first used by the American Nobel microbiologist Joshua Lederberg. So interest in this whole area kind of waned, particularly with the introduction of antibiotics and the whole scene. And also because it was so difficult to deal with organisms in the intestine. You really couldn't quantify them. There are trillions of them, and even classifying them was extremely difficult, if not impossible. So a lot of things came together in the 1990s, leading to an incredibly renewed interest in the microbiome, in this case of the intestine. And it was greatly added, as we'll hear, by technical developments. The ability to identify the 16S RNA as a means of initially detecting populations and then coupling that with more advanced forms of proteomics, of metabolomics, and even of individual sequencing of specific genes from the microbial population. And that sort of made technology lead to the explosion, because things are possible now in the course of a day that before might have taken a lifetime. And so this entire area has come into its own, and its relationship to metabolism, to nutrition, to inflammation, to disease, to cancer, the list goes on. And the evidence in some areas is strong and experimental, in others it's weak and hypothetical. And so to try and steer us through this sea, we're very fortunate in having two colleagues at NIH who are expert in these in this area. I'm sure that along the line there'll be some discussion of probiotics and of the treatment of particularly Clostridium difficile intestinal infection by fecal transplants. Something that initially took place in 1958 when a surgeon, Ben Eisman, gave a fecal transplant, that is, put feces from a normal individual into the colon of a patient with severe, I guess it was Clostridia difficile pseudomembranous colitis, and the patient got better, which is what happens to most of those patients today. So this is a whole amazing area. So our first speaker is going to be Yasmin Belkaid. Both she and Warren Strober are from NIAID. Yasmin took her PhD at the Pasteur Institute, came here for a while, went to Children's Hospital in Cincinnati, and then came back to the NIH, where she is in the branch, is it, or laboratory of, at any rate, explain. It's the same place where Warren Strober is. So now Warren has spoken before in this course, and he's one of the true experts on mucosal immunity, and his field is that of inflammatory bowel disease. Warren's been highly honored and is a distinguished senior investigator in the same laboratory. So Yasmin, maybe you would like to begin. Thank you.
Hello, can you hear me? Yes. It's a great pleasure to be here. Thank you very much for inviting me to give this lecture. I'm going to try to just talk to you about some of the general concepts that have emerged over the last few years in the context of the crosstalk between the microbiota and human health. And I'm sorry because I'm a little bit sick, so my voice may be a bit... So really, over the last few years, it's very difficult to open a magazine or any kind of journal without having a highlight of the microbiota and the role of this microbe on human health. And this really has taken central stage, not only in the context of the scientific community, but also in the context of the public. I think there is a growing awareness of the importance of the microbe that live in all the different surfaces of the body and the capacity of the host to maintain its host physiology. And I'm going to describe with you today some of those main roles in the context of both immunity, but also in the context of pathology. So coming back a bit to history, I think I'm going to go a bit deeper in history and actually a bit earlier. In fact, the description and actually microbe live in the different surface came earlier than Mechnikov. It was actually discovered that this microbe live in the oral cavity, and there was this beautiful describing here that you can see this nice illustration that was done in the 17th century. And you can see here in 2011 a fish imaging of the microbe of the oral cavity again. So early in the 17th century, we already knew that this microbe were actually present in our own body, and the mouth is actually colonized by a very diverse population of microbes. Coming back a little bit later, we actually had a German scientist that really had very seminal observation by the discovery that lactobacilli was quite important to the maintenance of the vaginal ecology. And actually the presence of the microbe were actually correlative and associative with the health of women. And later on, of course, we had this description earlier. We had the beautiful work by Mechnikov that didn't go completely right. He actually thought that the good microbe that we're actually drinking were replacing the bad one we had in the gut. But nonetheless, he was really the first one to actually link microbe and microbe that we can actually ingest with health. So it was really seminal discovery that really led to, of course, the discovery of probiotic and also the understanding that we need to maintain a healthy relationship with this microbe. And of course, we forgot all of that. Over the last few years, there has been an explosion of discovery and sequencing that has actually led to really the growing understanding that we have really a colonization of microbe in all the different body surfaces. And what's really extraordinary is the fact that each of these surfaces is really colonized by a unique population of microbe. And actually, there is a high degree of diversity in the context of one single tissue. And I will come back to this concept later. So all the different surfaces of the body are covered with highly diverse and highly specific microbes. It is believed, for example, that in the GI tract, we have over 1,000 different kinds of species that can actually colonize individuals. And the same number is actually true for the skin, for example. So as I mentioned before, all the different surfaces are actually colonized by microbe, but really how this microbe interact with the tissue is really not very well understood. Because in each of these environments, the microbe are going to establish a unique mode of interaction with the tissue. For example, in the GI tract, and I'm going to come back to this point later, this microbe live really essentially outside, and actually we are protected by a mucus layer. In the skin, these microbes are going to be present in really other structures. They're going to be inside the hair follicle, sebaceous glands. So really the way by which those microbes interact with the tissue is going to be central to explain how these microbes are capable to control the physiology and the function of each tissue. But really very little is understood really about how this is happening. So over the last few years, we have now finally understood that we are really a minority in our own body. And we are really a kaleidoscope, a composite of microbial community and human cells. And there is actually an estimation that there is 10 times more microbial cells in our own self than actually human cells. And at least 100 times more genes expressed at any given time by the microbe by our own genome. So really, we are really basically just an envelope carrying those microbes around, and we are of very little importance. So this is really what we are. We are a meta-organism, a composite of microbial and human cells. So we have to think about the microbe not really as these enemies. We are really home to these microbes. And all those different surfaces are really the natural habitat by which those microbes can survive. So they do something in exchange for us, of course. This is symbiosis. And what they do is many different things. The microbes that live in the GI tract and in other surfaces have been shown to be extremely important to control, for example, digestive system. They can actually help us digesting elements that otherwise will not be able to fragment. They can, of course, help the development of the brain, and there is actually fantastic work showing, at least in mice, that you can change anxiety by actually changing the kind of microbe you have in the GI tract. There is actually some fantastic work also highlighting the role of the microbe that live in the GI tract and testosterone level, and showing also that actually sexual hormones can be controlling also the microbiota. So very interesting interplay between hormones and the microbiota, and also between development and also in the terms of behavior and the microbiota. At the moment, all of that has been done essentially experimentally, but this is really fascinating to think how this actually play a role in the context of human health. There's, of course, a really fantastic work that has been done in the context of metabolism, and our experiment it has been shown that type 2 diabetes, for example, can actually be amplified by the presence of certain microbes. Also, beautiful work done by Jeff Gordon has actually highlighted the fact that certain microbe can actually change weight. For example, obesity can actually be transferred by certain kind of microbe. And really, you can experimentally do analysis by actually taking the microbiome of someone that is lean or obese and actually transfer the phenotype to an animal just by transferring the microbiota. And more recently, some work that was done by the same group has actually looked at twins and looked at actually obesity and actually was able to find associations between defined microbiota and weight gain. So I think fascinating correlation between metabolism and obesity, and also all the disease that actually link to this kind of obesity behavior. And of course, there is a very important work of the microbiota in the control of the immune system. I'm going to come back to that later. The microbes that live in the GI tract or other tissues are extremely important to maintain the capacity of the host to develop immunity to infection, but also are very important to allow the host to develop proper immune sites. So for example, the microbes that live in the GI tract are very important for the education of the mucosal immune system. And in absence of the microbe, you're actually going to have a reduced size of organs that are linked to development of immunity. So really, these microbes are important for the development of the immune system and its function. And really highlighting some of the discussion that was actually started earlier, there is really a fascinating discovery recently, and rediscovery, in fact, of the importance of this microbe by really the seminal observation and work that has been done by the clinician by, again, doing fecal transplant, taking patients that have C. difficile infection that have been actually not be cured by various treatments, and being able to actually transfer the microbiota from a healthy individual has been shown to actually be sufficient to treat patients that had lived for years with those chronic infections. And of course, this is something that is now a dream in the context of IBD and other disease. There is no proof that it's actually working yet. But this opens really an extraordinary possibilities, and actually changing the microbiota of someone is possible. And you can actually, in this context, treat disease in the context of infectious disease, but maybe other metabolic syndrome. So, and how this is working is not completely understood. I'm just going to show you one of the examples. One of the mechanisms by which actually the microbiota can help controlling disease is, of course, to competing with the microbe, because, of course, they're going to use the same space. But the microbe can also directly act on the immune system. And I'm going to give you one illustration here. In the GI tract, for example, this microbe contains ligands, in this context, some elements in the DNA, that can directly stimulate antigen-presenting cells in a way that actually allow those cells to really induce effector response against the pathogenic invasion and to block excessive regulation. And these together really allow this microbe, just because they have those ligands that stimulate the immune system, to really promote effector response against infection. And this really altogether allow the system to mount adaptive immunity to pathogen, but also response to vaccines. So really, these microbes that live in the GI tract can act as our primary adjuvant. They're really there to really promote and educate the immune system. It's another example of the positive role of the microbe that was done in our lab recently, in which we actually find that in the context of an acute infection, the microbes are actually capable to educate the cells that enter in the tissue to adopt appropriate function. So in this model of infection, most of the cells that are recruited in the tissues are inflammatory monocytes. And these inflammatory monocytes that enter in the tissue have the capacity to produce large amounts of prostaglandin. And what actually John was able to show is the fact that this prostaglandin was actually produced in response to the encounter with the microbiota, that was actually able to educate the cells that enter in the tissue to produce this molecule that in turn suppress the activation of neutrophils that create tissue damage. So this is just an example to show you how those microbes can act in very diverse way by educating the immune system, but also by inducing the appropriate mode of regulation in the context of mucosal inflammation. And this is also another illustration done at the NIH recently by the group of Giorgio Trinchieri that really beautifully illustrate the importance of the microbiome in human health, but also in the context of therapy. So what actually his group was able to show is the fact that if you actually have an animal that has a tumor, and if you actually treat those animals with antibiotics, or we use germ-free mice, so those animals no longer have actually any kind of microbial stimuli, these animals are no longer able to respond to immunotherapy in the context of this treatment. So this was actually a beautiful illustration that you need the microbe in the GI tract or in other sites to allow the host to respond to chemotherapy or immunotherapy. So this actually raises another issue, another important point, that in the context of treatment, we also in some cases need to preserve this microbe because they can really serve as adjuvant to promote immunity and clearance of those tumors. And this is just an illustration and one figure of this paper where you can see here that actually if you treat the animal with this kind of immunotherapy, you can actually abolish most of the protective effect by having those animals under antibiotic treatment. So really, you really need the microbe in this animal to actually be able to mount responses to this treatment in the context of tumor. This has, of course, really very, very important clinical implication. So to go back to this idea that the microbe colonizes different kind of surfaces, I'm going to illustrate some of the effect of this microbe by looking at another site. So most of what we know today about the crosstalk between the human health and the microbiota really was developed in the context of what we know about the GI tract. And of course, this is a site that has the highest density of microbe. But all the different tissues are colonized by unique and site-specific microbe. So I'm going to discuss with you some of the findings in another tissue, the skin. So the skin is a very large tissue. It's supposed to be probably the largest organ of the tissue, although this is debatable. This is a primary site of infection. It's also a site by which some vaccines are delivered. And it's also a site by which various inflammatory disorders can develop. It is also a site by which you can develop cancer. But the skin, really surprisingly, is also the site of a highly diverse population of microbiota. And this was really highlighted by the beautiful work of Julie Segre a few years ago at the NIAID that really illustrated the fact that the human skin was actually a composite of a large number of microbial community. And in fact, then you had probably the highest diversity of microbes in the skin than in any other tissue. And you not only have bacteria, but you also have fungi and also viruses. The microbes that live in the skin live in very unique structures. They're going to live in the hair follicle or sebaceous gland. And you can see here some image when you can see the fungi along the hair follicle. So this is actually a survey that was done by Julie's group a few years ago. And you can see here that what she was able to show is that all the different body sites that were collected have, in fact, unique microbial community. And there is a really high degree of diversity in each of the different sites that were collected. And this is relatively conserved between individuals. So each body habitat is actually hosting a very unique composition of microbes. It's actually a survey that was done more recently in the context of fungi that, again, illustrates this different diversity in human body of the different fungi communities. So when you think about a tissue and if you think about the microbiota, don't think about one kind of microbe covering all the surfaces, but each of these different microenvironments is really hosting a unique microbial community. So a few years ago, a student in my lab decided to address the potentiality that the microbes that live in the skin could also control tissue immunity. And she was actually utilizing germ-free mice, and I'm going to come back to that later, and was able to show that if you look at lymphocytes that reside in the skin, and they can produce inflammatory cytokine that can actually help controlling infection or different kind of settings, if you use a germ-free mice, there is a reduction in the capacity of the cells in the tissue to produce those inflammatory mediators. So she was able to show that in absence of any kind of inflammatory settings, if you don't have the microbe in the skin, your immune tissue is actually less capable to fight infection. And she was able to actually confirm that using an infectious model. So one tool that is utilized very much by the community that works on the microbiota is actually germ-free. Germ-free, we built a few years ago a germ-free facility in the campus, and this facility allows us to study the effect of defined microbe on the immune system because these animals are raised in complete absence of microbe. They are in bubbles. So we utilized a parasite infection that actually infects the skin. And we look at the capacity of the microbe in the skin to control the capacity of the host to mount protective immunity against this infection. So what you can see here is if you infect those animals with this parasite, they will develop a lesion and inflammation. But the first interesting observation, and if you do that in a mice that has no microbe on the surface of the skin, they are no longer able to develop inflammation. So this actually highlights that the inflammatory process is really not due to the pathogen, but often really due to this microbiota that is really living on the surface of the skin that really amplify the inflammatory response that is present. So if you do not have the commensal, you don't have pathology. However, not having inflammation is sometimes a bad thing because the immune system utilizes inflammation to develop immunity against infection. And indeed, if the animal do not have commensals, the T cells that arrive in the tissue are no longer able to produce the amount of cytokine that is required to control the infection. So the microbiota play a dual role. One is actually promoting the pathology, but on the other hand, it also helps us mounting protective immunity against these invading microbes. So this is actually an experiment to try to prove the link between the microbe and the immune system, and it's an add-back experiment. So we had used an animal that was completely empty, that had actually no microbes. So we decided to add back one single commensal. And this commensal that we decided to utilize is Staphylococcus epidermidis. We have actually a world leader on this microbe that is actually in this floor, on the building 50 floor. And we decided to add back this microbe to see if one single commensal microbe was sufficient to restore the capacity of the host to mount immunity against the infection. What you do see here is actually the mice have, again, if they are raised in the normal setting, they're going to develop an inflammatory responses. They have an immunity against the parasite. The mice are germ-free. They do not have immunity against the parasite, and they do not have a good T-cell response. If you add one single commensal in these mice, you can restore the capacity of the mice to develop immunity and to control infection. So I just give you this little story as an illustration of the profound role of the microbe on the immune system. And you can actually, in the complete absence of this microbe, have a profound immunosuppression, but you can actually restore the capacity of this microbe to develop immunity by adding one single microbes in those empty animals. So really, without this microbe, there is, in fact, no immunity and no capacity to develop protective response to infection. So again, they play a positive role in many different aspects. And they are, of course, these important partners of all different host physiology, including immunity, endocrinal level development, structural tissue. They also reinforce barrier. But this actually is not always a positive element, because what we also know is the fact that bare tissues, that are the tissue that are invaded by those microbes, are also the one that are actually the most often affected by inflammatory disorders. And over the last few decades in westernized country, there has been an incredible rise of disease that affect those bare tissues. And what you have to understand is all these tissues that are colonized by this microbe have extraordinary pressure. They have to maintain this huge microbial burden while at the same time in the GI tract really tolerate food and being exposed to pathogen. So this tight balance in this tissue can be very easily disturbed, and this is really what happened and is believed to happen in the context of disease such as IBD, and we're going to have a world leader in this field talking to us about that later, but also in the context of food allergy or in the context of asthma. In all of the different settings, it is believed that the incapacity to regulate responses at mucosal barrier or at surfaces will lead to those disorders. And really, a disruption of our relationship to the microbe is believed to be one of the very severe, important factors in the etiology of this disease. And this is a really alarming study that has actually looked at the number of actually antibiotic treatment given to children and the development of IBD. And as you can see, there's a dramatic correlation between the number of antibiotic treatment given to a child and the amount of disease that they can develop. So really, this is something that's happening right now. This is something that's happening in countries in which we overuse antibiotics. And there is really a dramatic increase also of allergy and asthma in these children. So because we have probably disrupted this relation with this microbe dramatically, there has been another side of the coin, which is, of course, the fact that this microbe are believed to also be agent of pathogenic states. For example, it has been actually shown that defined microbe can actually promote tumor development. The first example was, of course, H. pylori in the stomach, but actually there is more recent evidence that colon cancer may actually be also triggered by defined microbes. There is actually evidence that actually there is a bad crosstalk between certain microbes and heart disease. Of course, IBD is believed to be due to a disruption of the relationship with these microbes and can be amplified by the microbiota. There is, of course, the role of the microbiota in obesity. There is experimentally and also very nice clinical evidence that there is a link between the microbiota and various autoimmune disorders, but also in the context of other peripheral tissues such as the liver inflammation. So in all those different settings, most of them are supported by experimental data. There is a growing understanding of clinical association between the microbe that live in the GI tract or in other tissues and pathogenic states. So what have we done? So we have done quite a lot of things to disrupt our relationship with the microbes. Of course, antibiotics, which has been something extremely important for promotion of human health, has probably been one of the most important disruptors of these microbial communities. And really, this repetitive treatment of young children's antibiotic is really believed to be associated with very severe outcome, again, in the context of IBD or in the context of asthma. We also have changed our mode of living. I mean, are we more sedentary or are we doing extreme sport? Both of them have been shown to lead to change the microbiota. We, of course, have changed our nutrition in some places more than others. We have also changed our hygiene and the way we relate to the microbe in many ways. I think we utilize different kind of aggressive treatment. And importantly, we have changed also the kind of infection we are encountering. We used to have warm infection, for example. They were probably a very important buffer of the immune system. Today, what we have essentially is those acute heat of infection that are actually repeated and treated by antibiotics. So really, we have done quite a lot to change the way the microbiota are living in our different surfaces. And it is believed, and we have essentially a state of dysbiosis, that is a dysregulation of the appropriate microbe living in the different surfaces. So what I'm going to illustrate with you again is an example of how we can disrupt our relationship with the microbe in the context of acute infection, because this is one of the factors by which this microbe can actually be affected and altered. So how do we deal with the microbe under normal conditions? So this microbe lives in the surface, but what you don't want is actually this microbe to translocate. If they start to just invade your body, you become septic. So there is a lot of mechanisms that are in place to really maintain all this microbe outside of the body and not inside. In the GI tract, it is believed to be due to the combined action of a physical barrier, innate defenses, and adaptive responses that together create what has been referred by Andrew McPherson as a mucosal firewall that together really prevent the access of this extraordinary dense microbial population to the host. When this microbe can actually translocate, it is believed that regulatory T cells of various sorts are here to limit exuberant response to this microbe, because really everything is in place there to limit excessive reactivity to this microbe or translocation. And the combined action of all those different elements, both immune and structural, is creating what has been also referred here by Laura Hooper as the demilitarized zone that really is an empty space. So this is the epithelial cells in the gut. This is actually a fish staining of the bacteria. And this black area here is really a place where the microbes are really mostly not living. And they're not living because we have a mucus, we have IgA, we have antimicrobial peptide. And all those elements are in place really to prevent access of this microbe or too much access of this microbe to the immune system. But of course, things can change a lot. And that can change because we have antibiotic treatment or because we have infection. In westernized countries, it is believed that there is a minimum of 2 GI infections per year per children, which means that on a regular basis, this healthy relationship with the microbe can actually be challenged and can actually be affected. So we decided to look at what happened to this mucosal firewall and the separation of the microbe when you have an acute infection. So this is one kind of infection, and you can see again, similarly to the image I have shown before, then in a normal animal, in steady state, in the small intestine, you have a separation, and you can see that the microbes are not in tight contact with epithelial cells. 7 days post-acute infection, here due to Toxoplasma gondii, you can see an increase of the microbial community that you can see in higher density, but also then you can see that these microbes now are in tight contact with the epithelial cells. So one acute infection can change completely the way we relate to the microbes that increase in density and can also now become more tightly adherent to the epithelial structure and become more inflammatory. This microbe can also translate when you have an acute infection, and they can be found not only in the gut, but now can be found in the deeper structure. You can see here then some of the microbe can be found directly in the villi of the gut, or you can start to grow them from peripheral tissue. So there are actually a lot of instances where the microbes are no longer contained by this mucosal firewall, but can actually invade the tissues and can actually be found in the spleen or in other sites. The other thing that happens when you have an acute infection, as well as in the context of antibiotic treatment, is you change the kind of microbe you have. So for example, in the context of acute infection, it is actually shown by various authors, including us, then Proteobacteria, and in particular gamma Proteobacteria such as E. coli, can become very dominant. And it's actually what you can see here. If you look at the bacteria that are close to the mucosal barrier, you can see that in the context of acute infection, most of the microbes that are collected are in fact belonging to gamma Proteobacteria and E. coli. And these microbes that when they are present in small density are not very, very inflammatory, when they really become, the vast majority of the microbes in the GI tract become part of the inflammatory process. And what is really interesting is the fact that in patients that really suffer from chronic inflammatory disease in the GI tract, Proteobacteria also become a very dominant population. So when you change the kind of microbe you have, either because you have an infection, antibiotic treatments, or you have a different kind of diet, you create a pool of microbes that has actually more aggressive potential and that can amplify inflammatory disorders. So what I really would like to kind of finish with is really the fact that we have to constantly take into account the microbe when we think about human health. We are not living alone. We are a composite of microbial communities and ourselves. And devising actually strategies to just control a bare immunity or bare inflammation really need to take into account this constant dialogue with these evolving microbes. But there is a lot of thinking that we have to do as a community of what we have done to this microbial community and try to think how we can actually learn to reestablish a healthy relationship with the microbe or try to reestablish the microbe we have eliminated from a microbiota. There is actually some hypothesis going on right now that what we may have done is we may have eliminated microbe, and there is actually people proposing that there is a disappearing microbe theory in which what we have generated is a microbiota that is not diverse enough to have the resilience and capacity to adapt to all the different changes. So on that, I'm going to let you, for our next speaker, I can take a few questions and have our next speaker that's going to talk about real disease, IBD, and how the microbe contribute or can be utilized in the context of this inflammatory disease.
Is it on now? Yes. Switch. So you've shown very nicely the consequences of dysregulation of the microbiota. It's not clear to me exactly what we're supposed to do to keep it the way that it's supposed to be. So what's your recommendation?
I will make absolutely no recommendation at this stage. But I think what is important to understand is we have a state where we have an extremely emergence of inflammatory disease. I mean, most children develop allergies at extraordinary rate. We have clearly done something that is actually fundamental in disrupting those healthy relationships with the microbes. And we have now realized also as a community that these microbes are extremely important. So I think it's a stage of reflection about what have we done. I mean, have we overused antibiotics? Do we need actually more specific antibiotics to actually stop altering our entire community? Can we restore appropriate microbiota? I mean, in the context, I think the example of what happened with C. difficile and fecal transplant is very, very informative. Then there is maybe hope. Then if someone has actually chronic inflammatory disorders, we may be able to transfer a more appropriate microbiota. But there is a lot still to do. How do we actually make those microbiota populations take?
And I know there's a general theory that we're just trying to be too clean. We're trying to take care of our kids too much. We should let them eat dirt and play with pets and stick stuff in their mouth and do all that kind of stuff. Do you think that's about right?
Well, I think it's an extension of the hygiene hypothesis, which really that postulated that we need to actually maintain exposure to microbial product or microbes to just be healthy. But I think it's a composite of things. It's maybe not just hygiene. I mean, we have also changed dramatically our diet. We have also, a lot of treatment with antibiotics that may not be always required. I mean, antibiotics are extremely important, but I think they may have been overused. It was a very nice talk last week by Marty Blazer at the NIAID that really highlighted that in different parts of the country, culturally, people use different amount of antibiotics. So, for example, certain part of the country use more antibiotics than other places. There's a lot of cultural issues around how we relate to those treatments. But I think what is important, we're not going to be removing the antibiotic from the equation. But what can we do in the context of a treatment to make sure that we actually follow up and add back elements that may have been disrupted, or actually utilizing strategy to minimize the impact of these antibiotics is one of the things. But I think there is more than that. Another element that is important is what I refer to the worms infection that used to be our partners until very recently in life. We used to be chronically infected with worms. And these worms were probably our first shield for this adjuvant effect of the microbiota. Removing them from the equation may have led us with sensing this microbe maybe too intensely. So there's a lot of reflection in terms of the community. We're not going to introduce the worm either, but can we learn from what they have done to try to just minimize the inflammatory impact of the microbe?
So, Yasmin, would you say something about the technology that you use to identify components of the microbiome and some concept about sort of how long does it take to actually do an examination say from a human fecal sample or something like that? And to what extent do you then do genomic sequencing from it? How do you select what you're going to look for? Is it all done from examining gene sequences of things that are likely to be? How do you select what you're going to examine?
So it really depends. I think people are moving away from just making a catalog of the different species to try to look at what the microbes are doing. Really looking at gene expression is actually where the things is happening. And more than that, there is actually also evidence that different isolate or do different things. So I think the field is also growing to look at the function of the microbe rather than just making a catalog. I think it's been very informative, trying to understand what kind of microbe are there. But the most important part is actually how they're adapting to different metabolic states and what genes they can actually upregulate or not. And I think that's what is the next step in our understanding of how those microbes modulate human health.
I'd like to ask, how does this current theory explain the fact that 100 years ago, life expectancy was about 40 years, and most people died from overwhelming infections?
Oh, no. I mean, I think we live sick very long time. We live with disease very long time because we have a fantastic healthcare system that helps, that people have chronic infection. I think people live very long time with chronic infection. We have found ways to make people surviving with those chronic infections. It still doesn't mean that it will be an acceptable way to live with this chronic situation. So yes, life expectancy is longer because we have antibiotics, we have better hygienes, we have removed certain infections, we have vaccines, but at the same time, we also live with very severe disease for a very long period of time that could be potentially avoided.
So when people go into CVS, I'm sorry,
So Yasmin, since sometimes antibiotics and even very broad spectrum antibiotics are necessary in life-threatening situations, has anyone done studies in humans looking at how long it takes to recover the microbiome, particularly the gut microbiome, after a course of broad spectrum antibiotics, and how much of that is reconstituted from residual endogenous bacteria and how much from other sources, and to what extent one can influence that by giving probiotics or other sources of bacteria either during or immediately after antibiotic treatment?
So what was actually quite remarkable when people did those studies, and they're still doing those studies, and there is actually growing knowledge about that. What is interesting is not everybody recovers the same way. So you're going to have some people that can rebound and actually kind of recreate the diversity, but some that can't. Why some people can, or how much the genetic factors or the kind of microbe you have explain these differences is not very clear. But there are actually people that will never recover. So I think you're absolutely correct, and I think learning from what we could potentially add at certain times in terms of maybe metabolites, because remember, those microbes actually basically are going to, in an ecological system, we may be able to give back certain metabolite, to give back the capacity of the microbe to compete in this kind of situation. It's terrible inflammatory settings for them. I mean, they can actually become, and that's in the context of C. difficile, emerge in the context of sometimes one course of antibiotics.
Any other questions at this point? Well, save your questions. We can discuss more after Warren speaks.
I'm going to approach this topic in a little bit more specific way by using a specific disease, Crohn's disease, which is a kind of inflammatory bowel disease, to use that as a microscope to discover some of the aspects of the microbiome and how it interacts with the body and how it sometimes is involved in disease processes. Crohn's disease, very, very quickly, is an inflammatory disease of the small and large bowel, and it's a chronic disease. It can cause fistulations, which are passages to other structures or even the skin. It can cause strictures. It can cause abnormal bleeding and diarrhea. And it can cause a lot of extra-intestinal manifestations as well, including uveitis and skin disease. Patients are treated either with surgery when they have severe strictures, or they can be treated with steroids, or more recently with anti-TNF type of therapy or other kinds of anti-cytokine therapies. And this is an example of the histology of Crohn's disease. You can see a lot of granulomas in the tissues. You can see in this barium enema, you can see a stricture, you can see the skin manifestations, and so on. This is a very brief overview of this rather common disease. Everybody seems to know another individual, either a friend or a relative, who has this disease. Now, there's a consensus about what the disease is caused by, and the idea is that the gut mucosal immune system responds to either pathogens or commensal organisms, as Yasmin pointed out, with immune responses that are either protective or they can cause inflammation. And in the case of inflammation, they produce a lot of interferon gamma and IL-17 and TNF-alpha, which are pro-inflammatory cytokines which mediate the disease. Now, we think that the microflora or microbiome is very important in inducing or initiating, sustaining this inflammatory reaction. Because there are numerous models of gut inflammation, and they all are either treated by or can be prevented by a germ-free notobiotic environment. In fact, you can even treat certain patients with antibiotics and get improvement. So there's no question that the gut microbiome either contributes to this disease or is a major driver of this disease. Now, there have been numerous studies of the microbiome in Crohn's disease, and I'm just going to summarize it very, very quickly here for you. There's quite a bit of disagreement about the changes that occur, and the statements I'm making here are more or less agreed upon. The firmicutes, which this is the name of a phylum, is depleted, and Proteobacteria, which include E. coli organisms, are increased in a subset of patients with Crohn's disease. And mostly, this is limited to changes in the small bowel of patients rather than the large bowel. The bacterial DTs are either decreased or increased depending on the paper that you read. And an organism called Faecalibacterium prausnitzii and Clostridium species are decreased. We're going to come back to that. This is a very important point. There's no evidence that the disease is caused by a pathogenic organism. So these are all commensal organisms that ordinarily don't cause disease. And there's a decreased bacterial diversity. Now the question is, how do these changes that are admittedly are occurring only in a fraction of the patients, not all the patients, how are these changes affecting the disease? Are they primary changes? Are they actually causing the disease, or are they secondary changes? And if they are secondary, do they aggravate the disease? We'll try to answer some of these questions as we go along. The first part of my talk will be the microbiome and gut inflammation. This is a more general discussion of the microbiome in relation to gut inflammation. Now, the first thing we have to understand is that, as Yasmin pointed out, the microbiome can be very protective, can help. And we know from several studies that if there is not too much break in the barrier, the epithelial barrier, the mucosal mucus barrier here in the gut, the microbiome can stimulate dendritic cells to make various kinds of regulatory cells which dampen immune responses. These can either be FOXP3 positive T regulatory cells or FOXP3 negative LAP positive. LAP stands for TGF-beta positive cells. This occurs when the breaks are very minor. So not very many organisms are coming in, and yet they can induce a regulatory response. On the other hand, if you induce very large breaks in the barrier, such as you see in certain kinds of experimental colitis, for instance, dextran sulfate colitis or DSS colitis, this mechanism becomes overwhelmed by a positive immune response, the effector cell response, which causes inflammation. So this is your first line of immunological responsiveness, a protective regulatory response. Now, we know now that there are different kinds of organisms, as Yasmin pointed out, and there are organisms that can particularly induce these regulatory responses, and there are other organisms that can induce more inflammatory responses. There's been a great deal of study, particularly of clostridial organisms, and also of Bacteroides fragilis. I'm not going to talk very much about Bacteroides fragilis, but it's in the same category as these organisms. And there have been a very brilliant set of studies by Dr. Kenya Honda in Japan at the Riken Institute. He and his colleagues have done really splendid work on the role of clostridial organisms. And what we see here is that Bacteroides fragilis can induce regulatory cells, and clostridial organisms can induce regulatory cells. Other organisms, particularly in the small intestine, SFB organisms, can induce Th17 effector cells. These effector cells are important in protecting us from infection, or as Yasmin alluded to, they can also be involved in autoimmune disease, particularly arthritis. Now, what they did, what the kind of study that Kenya Honda did, was to take fecal material, human fecal material, and colonize germ-free mice with human fecal material. And by looking at the kinds of organisms that would induce regulatory cells, they were able to narrow down to which organisms were actually inducing the regulatory cells. It turned out to be clostridial strains. They found, first of all, that organisms, if you took fecal material from the cecum of humans, and you treated them with chloroform, the ones that were resistant were more capable of inducing regulatory cells. And they then went on to, without going into the details, they went on to narrow down which organisms were actually inducing the regulatory cells. It turned out that they were not primarily, but almost entirely Clostridial organisms. So they developed the theory that Clostridium, in particular clusters, can induce dendritic cells, CD103 positive dendritic cells, to act on naive T cells and induce T regulatory cells. And these blocked the effector function of other cells and therefore prevented inflammation. Before I go on to this slide, I want to say is that this has led to a very interesting possible therapy that plays upon this idea of fecal transplants. Because instead of giving an entire fecal transplant to treat a patient, it now becomes more rational to get certain subspecies of organisms, as in this case, Clostridial organisms, and give soups containing Clostridial organisms and presumably prevent inflammation in that way. We often think of Clostridial organisms as causing disease, and in fact, Clostridium difficile does cause disease. But these are other Clostridial organisms, and these induce regulatory cells which actually prevent inflammation. I'll come back to that in a certain way a little later on. Now, I want to make a distinction between organisms that can cause, disease. We think of colitogenic organisms, and there's only actually one example of colitogenic organisms. I'll tell you about that. These are organisms that, present in the gut lumen, can cause inflammation. And then there are other types which don't cause inflammation themselves. These are actually colitis promoting or colitis inducing, colitis helping organisms. If there's another reason for the colitis or the inflammation, these will make things worse. Now, let's start with this colitogenic organism. This was discovered by some very brilliant studies by a woman named Wendy Garrett at Harvard, working with Lori Glimcher. And they developed a model of colitis called the truck mouse. A very complicated mouse. They had deficiencies of an important transcription factor called T-bet, and they were also RAG deficient mice. And it turned out that if you lacked T-bet, the macrophages produced a lot of TNF, because T-bet is a negative factor for the production of TNF-alpha. So they overproduce TNF-alpha, and they cause inflammation. That was the origin of the inflammation in these truck mice. But at the same time, because they were RAG deficient mice, they also didn't have T cells. And because they didn't have T cells, they couldn't make T regulatory cells, which I talked about before, as preventers of inflammation. So they had a double whammy. They made too much TNF on the one hand, and they didn't have regulatory cells on the other. That's why they developed a lot of inflammation. Now, we wouldn't have been that interested in this model, a very complicated model, except for one thing. What Wendy Garrett discovered is that if you took normal animals and co-housed them with these animals, the normal animals would develop colitis as well. Or you can take normal pups and use these animals as mothers, and the normal pups will develop colitis. So this was either vertically or horizontally transmitted colitis. Then these normal animals did not have disease, and they had no reason to have disease. If you had not co-housed them, they would not have developed disease. There was no other pre-existing condition for them to develop disease. It was only the exposure to these organisms that led to disease. Well, Garrett and her coworkers spent a lot of time trying to figure out which organisms were actually causing the disease. They eventually settled on Klebsiella and Proteus. You had to have both Klebsiella and Proteus species to develop these diseases, and you actually had to have them in the context of a normal flora. That was never figured out. If you took the purified Klebsiella and Proteus and gave them to a normal animal, the normal animal would not develop disease. You had to give these in the context of a normal flora. We haven't figured that out yet. There was some subsequent work suggesting that it wasn't due to Klebsiella and Proteus, it was actually due to a Helicobacter species. So there's still some question about this, but it raises a very important theoretical question. That is, can you have an organism in the gut which will cause de novo disease in a normal animal? And if so, is there such an organism existing in Crohn's disease or other forms of inflammatory bowel disease? Now, interestingly, if Wendy treated the animals with agents that cured or prevented the inflammation, such as anti-TNF, as I mentioned to you, anti-TNF causes this inflammation. You treat them with anti-TNF, you can cure their disease. When you cure their gut disease, their immunological disease, then their flora no longer has this cholinogenic organism in it, and they cannot transfer disease with their stool content. So that, although these organisms were chlinogenic, their development in the gut depended on the presence of a pre-existing inflammation. That suggested that in an ordinary situation where you don't have inflammation, these organisms would not develop. Now, we get on to the second kind of organism, the organism that doesn't cause inflammation, that causes inflammation without gut disease, to one that makes other pre-existing gut disease worse. This is a study of mice with NOD2 deficiency. Now, you're going to hear much more about NOD2. I'm going to talk about that and how that functions. NOD2 is a intracellular innate immune factor which recognizes a component of peptidoglycan, part of the bacterial wall of bacteria. And what you see here this is a complicated slide, and I'm just going to say very quickly that the NOD2 deficient animal develops more severe DSS colitis. When you give them DSS, they develop more DSS colitis than a normal animal. So they have a pre-existing condition for making them worse. Now, the remarkable thing is that if you co-house, these are wild-type animals, and you co-house them with normal animals, they have a certain amount of DSS colitis. But if you co-house a wild-type animal with an animal that has NOD2 deficiency, then the wild-type animal will develop more disease. So this is the same situation as before, that exposure to a microbiome from an animal with NOD2 deficiency will make a normal animal have worse disease caused by something that causes disease in the first place. And remarkably, this is a look at the kind of microbiome that develops in these animals, and you can see that there are less firmicutes, that's in blue. There are less firmicutes in the animals that were co-housed with the NOD2 animals than in the animals that were co-housed with normal animals. So one of the things that's happening is that by exposing a normal animal to an animal with NOD2 deficiency, giving both of them DSS colitis, you will get a different flora in the normal animal. And this flora, to some extent, resembles that seen in Crohn's disease. Now, I mentioned already, this is a repeat of a slide, that there are organisms that can induce regulatory cells. That's the Clostridium specifically. But there are other organisms that have been implicated as inducing inflammation. And a good example of that are adherent E. coli, adherent invasive E. coli, AIEC. This has been studied particularly by French investigators. And the idea is that this AEIC are found in the colon, in the small intestines, not in the colons, in the small intestines of patients with Crohn's disease. And it's been postulated that these AIEC are sort of semi-pathogenic. They can penetrate to some extent, they can get into macrophages, they can live in the macrophages, they can induce the macrophages to produce inflammatory cytokines. They adhere to the surface of the epithelium by binding to a particular protein called CCAM6. Turns out that CCAM6 is induced by the inflammatory process itself. So the idea is that these organisms are causing Crohn's disease, but they're only present in some of the patients. Now, I've done a lot of reading about the characteristics of AIEC. I've been asked to comment on whether it actually causes Crohn's disease. And there are reasons to believe that they don't actually cause Crohn's disease. First of all, they're present in only a minority of patients. They adhere, as I said, via this substance called CCAM6, but CCAM6 is upregulated by inflammatory cytokines. So you have to have the inflammation first, produce the inflammatory cytokines, you induce the CCAM6, and now you can get binding of the organism to CCAM6. You need this binding in order to get the proliferation of these organisms. There's no evidence they can really enter the intact lamina propria. So they're really invading only in situations where you already have ulceration. So my view is that these organisms developed as a result of the inflammation, but once they develop, they can aggravate the inflammation, but they don't actually cause the inflammation. So that's what I'm going to say as a general rule about the organisms in the GI tract and how they interact with the gut immune system. In summary, some of them induce regulatory cells, and some of them can be clitogenic or semi-clitogenic. Now I'd like to turn my attention to NOD2. As I mentioned, this is a innate immune factor. It's an intracellular cytoplasmic factor. It reacts to uramal dipeptide. Uramal dipeptide is a small peptide that is derived from peptidoglycan, which is part of the bacterial wall. There's been a great deal of interest in NOD2 because NOD2, which is encoded by a gene called CARD15, a member of the NLR family of proteins, it is about 10 to 15% of patients with Crohn's disease bear polymorphisms of the CARD15 gene, which create loss of function abnormalities in NOD2. So the question is, how does NOD2 become a risk polymorphism? I call it it really shouldn't be called a mutation. A mutation occurs when it's only involved in diseased patient. A polymorphism occurs in a great deal of normal individuals as well. So you have to have other things besides the polymorphism to have the disease. There are many patients who are homozygous for this polymorphism, and yet they don't have disease. Now, the structure of NOD2 is shown here in diagrammatic form. There is a leucine-rich region, which is the area that actually recognizes muramal dipeptide, and there's one particular polymorphism called a frame-shift mutation or polymorphism. This causes a truncation of the LRR region. Since this region recognizes MDP, it's easy to understand that when you have these changes, there's loss of recognition and loss of function of the molecule. Other parts of the molecule are the nucleotide binding domain and the CAR domain. The CAR domain is very important for signaling function. There are also mutations in this region, and this gives rise to the Blau syndrome, and it causes not gut disease, but diseases of the eye, the skin, and the joints. Now, two competing theories have developed to explain why NOD2 polymorphisms would cause Crohn's disease. And both of these theories are based on how the body reacts to the gut microbiome. The first theory, I would say the dominant theory, because I think more people believe in this theory than another theory that I'll tell you later on, involves the fact that NOD2, in addition to being expressed in antigen-presenting cells, macrophages and dendritic cells, is also expressed in a very interesting cell that exists at the base of the small intestinal crypt. These are called PANF cells. PANF cells make a variety of antimicrobial products, shown over here. Lori Hooper, that was mentioned before, is very actively studying these. But one of the important products, addition that it makes, is a product called the defensin. Defensin is a relatively low molecular weight material, which can kill organisms. So it's part of the host defense system. These are defensin granules. Now, it turns out that if you have poor NOD2 function, or if you lack NOD2, as in mice with knockout of NOD2, you don't make very much defensins. For some reason that is actually poorly understood, NOD2 acts as some kind of factor that is necessary for the production of defensins. So the first theory is that because you have a NOD2 mutation or polymorphism, you make less defensins. Because you make less defensins, you don't control your gut microbiome. There's more proliferation of organisms in your GI tract, especially your small intestine where these Paneth cells exist. And because you have more growth of commensal organisms, there's more penetration of the gut. And now you overwhelm your regulatory system that I pointed out before. You make a effector cell response or an inflammatory response, and this is what causes Crohn's disease, at least of the small intestine. It doesn't work that well for the colon because there's less, there's no Paneth cells in the colon and there's less defensin production. Now, in another study, this was conducted by a woman named Salzman, you could show that in mice that lack Paneth cells completely, they lack Paneth cells, they get an altered gut flora. And in fact, notice that these are knockouts for a metalloproteinase that's necessary for defensin secretion. And they have less bacteroides. Actually, you have more firmicutes. These are the organisms that have the Clostridia. So this doesn't fit the theory that well because if you knock out defensin production, you actually have presumably more organisms that can induce regulatory cells. But anyway, that's the finding. And this led to this complicated slide, which I'm not actually going to go over. This led to people actually looking at how much defensins patients with Crohn's disease make. These are Crohn's disease patients who either have or do not have this NOD2 polymorphism. And in one study, which was very widely advertised, you might say, it was most people refer to this study when they're talking about this. Without going into the details of this, what they showed was that the patients who had the NOD2 risk polymorphism were making not no defensin, they were making less defensin in the small intestine than Crohn's disease patients who did not have the NOD2 polymorphism. Now, one thing that was a problem with this is that if the patients did not have disease of the small intestine, they had normal amounts of defensins, even though they had the NOD2 polymorphism. This suggested that even in these patients, the decrease in the NOD2 defensin was secondary to the amount of inflammation. And another study appeared several years later, doing the same thing with a larger group of patients. And in fact, in this study, which originated from Australia, there were no differences between the patients who had the NOD2 risk polymorphism and the other patients who didn't have, the Crohn's patients who did not have this risk polymorphism. So they found no relationship between the NOD2 polymorphism and the decrease in defensin. They said that all the decrease is due to increased inflammation and decreased Paneth cells. And in fact, experimentally, you can show that if there's a lot of interferon gamma produced, that has a negative effect on Paneth cells. So there is controversy in this area. One study saying there is decreased defensins, associated with this risk polymorphism, and another study saying there is not decreased defensin associated with this polymorphism. But again, the dominant idea is that if you have this NOD2 risk polymorphism, you make less defensins, therefore you have less host defense against commensal organisms. These commensal organisms enter the lamina propria and then ultimately cause disease. That is the dominant idea of how NOD2 risk polymorphisms work. But there's another idea. And that was originated in our laboratory and subsequently seen in other places as well. And that's what I'm going to talk about now. And again, this is a complex slide. But essentially what it shows is that if you pre-stimulate cells with MDP, the peptide that activates NOD2, you pre-stimulate the cells, and then you come back with a second stimulation with a TLR ligand, a substance that stimulates the cell through the TLR receptor. You can show that the pre-stimulated cell, the cell that had been stimulated with MDP, have a lower response to the TLR stimulus. And that's across the board. It didn't matter what kind of TLR stimulus. If you pre-stimulated with MDP, you caused a reduced response subsequently to the TLR, and that affected not only IL-12 responses, but IL-6 responses and other pro-inflammatory responses. Now, we found that as part of the response to MDP, the cells were making a substance called IRF4. This is an intermediate which has many roles in the immune system. It affects B cell function, affects dendritic cell function, it affects production of various cytokines. So it's kind of a jack of all trades. It makes a lot of it has a lot of different roles. I'll tell you about one of the roles in a moment. But this ability of MDP to prevent responses to TLR antigens led us to treat animals with MDP. So, and this is a study of TMBS colitis, a kind of experimental colitis. If you give TMBS, trinitrobenzene sulfonic acid, to mice, they lose weight, they produce a lot of cytokines, and they have inflammation. That's the blue line here. If you give them MDP first, they don't have the inflammation. Why is this happening? Because, as I said, the MDP is preventing the responses to the TLR response. There's a lot of organisms in the gut of these animals, and presumably those TLR responses are necessary to drive the inflammation, as I said at the outset. If you prevent these responses, you're not going to get the inflammation. We thought so highly of this idea, we actually patented it, but it's so far not been used to treat patients, but maybe someday it will be. Now, if you, this is another kind of inflammation, DSS colitis. It's also, if you give MDP over here intraperitoneally, you prevent the DSS colitis. But you don't prevent the DSS colitis if the animal lacks NOD2. You need the NOD2 to induce this regulatory response that prevents the inflammation. I'll tell you why you need it in a moment. It has to do with IRF4. Again, a complicated slide, but if you try to prevent colitis, DSS colitis in this case, in an animal that lacks IRF4, the MDP doesn't work. So that's telling us that in order to have this protective response, in order to what we call cross-tolerize responses to TLR ligands, the MDP relies on the presence of IRF4. How does that happen? I'm not going to do that one. Now, this is just a review of what I just said. MDP stimulates the cell to produce and stimulates through NOD2. That happened a little too fast. Then you get IRF4. The IRF4 somehow blocks the responses to various TLR ligands, and you don't get the inflammatory response. Now, how is that happening? This is material that we recently developed. And in order to explain how this works, I'm first going to tell you that when NOD2 is stimulated by MDP, or NOD1 is another closely related factor, is stimulated by its ligand, which is also derived from peptidoglycan, you interact with a protein called RIC, or RIP2. RIP2 is ubiquitinated. And this whole, and then it interacts with downstream molecules, in this case TAC1, which then activate the NF-kappa B and MAP kinase pathways. There's a lot of ubiquitination going on here. Ubiquitination is a process that activates various intermediate molecules. It's like phosphorylation, but it's a different chemical modification. And if we do complex immunoprecipitation studies, immunobinding studies, we could show now that if you pre-stimulate cells with MDP, these are pre-stimulation of the cells, the RIC, which is activated by NOD2, binds to IRF4, and that's shown with this band over here. If you don't pre-stimulate, you don't get binding to IRF4. But not only does RIC bind to IRF4, it binds with a number of other downstream molecules, in this case TRAF6, and with MYD88, which is very important in TLR responses. So one of the consequences of stimulating cells or pre-stimulating cells with MDP is to create a RIC that can bind to various downstream molecules that are important in the inflammatory response. And this also can be shown with this beautiful technique called dual-ink staining. This is a kind of staining that if two molecules are close together, they can develop a color, in this case a red color. And you could show that if you pre-stimulate cells, then there is binding between RIC and IRF4, and that shows up as a color, and also between IRF4 and TRAF6 and RF4 and MYD88. So this shows the same thing. Now, this has something to do with ubiquitination. Again, this is a complex slide, but if you take normal human dendritic cells and you stimulate the cells with MDP, you get ubiquitination. But if you, this ubiquitination is prevented by siRNA for IRF4. So this is RIC. This is IRF4. And the IRF4 you see here is, this shows ubiquitination. When you add IRF4, you get reduced ubiquitination. And this is true for it's a special kind of ubiquitination called K63 linked polyubiquitination. And this is occurring with not only with respect to RIC, but also with TRAF6. And in this complicated slide, we show that if we downregulate IRF4 with an siRNA, we prevent the decrease in polyubiquitination. So the long and short of this is that if you are pre-stimulating with MDP, and the MDP will bind to RIC, will bind to TRAF6, and then IRF4 will also be involved, and the IRF4 will prevent the polyubiquitination of the RIC or the TRAF6. Now, we took advantage of this, and we went back to the study where you can prevent TMBS colitis with giving the animal MDP, and this shows the same thing as before. This is TMBS colitis. If you give the animal MDP, they don't develop the colitis. They don't develop inflammatory colitis, shown histologically, and they don't develop various aspects of NF-kappa B activation. But in this slide, we show that when you give the MDP to the animal, there's a great increase in the amount of IRF4 in the tissues. And in fact, the IRF4, you could show the IRF4 is binding to RIC, it's binding to MYD88, and it's binding to TRAF6. These are the animals pre-stimulated with MDP. So this is happening in vivo as well as in vitro. In other words, by giving the animal MDP, you're reducing the binding of IRF4 to these various important intermediates that are necessary for NF-kappa B activation. And at the same time, shown over here, you're preventing the polyubiquitination of these factors. Now, you can go one step further. Instead of giving MDP to the animal, you can give the animal an IRF4 expressing plasmid, and you can prevent disease by giving the IRF4 expressing plasmid. This plasmid is labeled so you can identify it in the tissues. It's flag, so-called flag labeled. And you can show that the IRF4 that you give by the plasmid binds to these various intermediates, including MYD88, TRAF6, and RIC. So you can short circuit the system. Instead of giving MDP, you give the downstream molecule that actually causes the prevention of ubiquitination. And you can actually you can treat TMBS colitis by giving the IRF4 plasmid later on when the disease has already developed. The animal will start gaining weight over here. You can actually reverse the disease. So what we think is happening is that muramal dipeptide interacts with NOD2. The NOD2 then interacts with RIC, and the RIC interacts with IRF4. The IRF4 now deubiquitinates various molecules and prevents their activation. And I guess I went, that was the last slide anyway, but what does this mean in terms of the microbiome? It means that you have a molecule called NOD2, which responds to MDP. And that MDP is part of the microbiome because it's part of the bacterial wall of bacteria that's present in the gut. So the gut has a way of downregulating TLR responses. This is presumably very important because the gut microbiome could induce a lot of TLR responses and cause a lot of inflammation. That's where there's so much microbiome. This is, we think, an important mechanism of downregulating that response and preventing inflammation. If you have a mutation or a polymorphism that inactivates the NOD2, you will not have this regulation. You'll respond too much to your microbiome, and you will develop Crohn's disease. Now, to bring this back, which theory do you believe? Do you believe that the NOD2 is working through defensins, lack of defensins, and this is causing overproliferation of bacteria, penetration of bacteria of the gut? Or is it reacting? That would be an effector mechanism. There would be too much functionality there, too much response to the microbiome in that theory. The opposite is that there's not enough regulation mediated by NOD2. We think that it's the latter that's really the most important factor, and the reason we think that is that if you assume that there's too much response, because the NOD2 is not there, there's too much proliferation of commensal organisms. There should also be too much proliferation of pathologic organisms. The patient should have a lot of infection with pathogens, which they don't have. So we believe the second theory is the more valid one. Thank you.
Former life being the clinician dealing with patients about one recognizes that those are complex diseases and the search for organisms. Initially, the search for organisms that were pathogenic is now giving rise to this very complex interrelationship between the microbiome and the inflammatory response itself.
So there's some commensal organisms like lactobacilli that are thought to have anti-inflammatory properties and have been found to have anti-inflammatory properties in different models. Is it known whether there's a correlation between production of MDP and this anti-inflammatory property of certain organisms?
No. First of all, the use of probiotics, which very frequently contains lactobacilli, has been a big failure in Crohn's disease. We really cannot really effectively treat Crohn's disease by giving probiotics as we know them today. So I don't know of any relationship between any particular organism and MDP actually is more associated with gram-positive organisms. There's more MDP on the surface of than gram-negative, and of course there's more gram-negative organisms in the gut, but there are gram-positives as well. I don't know of any organism that has more MDP or something like that. There's plenty of MDP around in the gut as a whole.
Yeah. Lactobacilli lack LPS, but I'm not sure about MDP expression.
No, that has nothing to do with MDP. It has nothing to do with MDP. So, I mean, the future of probiotics may lie in these studies that I pointed out with the Clostridia. So if you have specific regulatory cell-inducing organisms, these may be very effective in treating or preventing Crohn's disease. And in fact, one thing I didn't tell you is that the same authors, Kenya Honda and his colleagues, actually in their supplementary material, this vast Nature paper that they published, actually 2 Nature papers, they showed that there was reduced numbers of Clostridia in the stool, in a fecal study, of patients with what they called ulcerative colitis. So they already have preliminary evidence that there's reduced amounts of Clostridia, and you know what relationship that has to either the cause or secondary effects of the disease. We don't know, but still, it's very, very provocative finding. One thing I didn't point out with that study, very interesting study, is that the way they think the Clostridia works is that it induces some short-chain fatty acid, which then induce epithelial cells to make TGF-beta. And the TGF-beta then induce the regulatory cells. Very interesting hypothesis.
What's the microbiome population of the normal small intestine in man? Do we know that?
Well, first of all, we know that there are a lot less organisms in the small intestine than in the gut, by a factor of at least 10. But in terms of the quality, this we do know because a very beautiful study by Frank and his colleagues have, studied the microbiomes of normal, large, and small intestine. There are differences. Maybe Yasmin is more acquainted with that than I am, but they all show a lot of firmicutes and bacteroidetes. Those are that and the Proteobacteria, the 80% of all the bacteria.
Two questions. The first question, with the animals where you have co-housing, how controlled is it in that, is it only within one cage, or do you have it across cages when you don't really want it?
No, they have to be in the same cage. They've got to be in the same cage, yeah, because they eat a lot of, they eat feces. So when you have animals in the same cage, they basically share the.
So even though the animal handlers might be changing cages or whatever, it's not being transmitted by the multiple, changing 20 cages at a time or something like that.
Conceivably it could happen, if they're sloppy, but generally.
But it hasn't been.
Yeah.
The next question has to do with where we are with the microbiomes of Crohn's patients in terms of someone who's in remission or not. Even though the probiotics haven't worked, their overall lifestyle and diet and antibiotics, there might be correlations with who is in remission longer and what their microbiome looks like versus another Crohn's patient.
Generally speaking, what people have found, there's not too much study of this, but what people have found is that when patients are very well treated, their microbiome reverts to normal. So again, it sounds like it's secondary to the disease process. The treatments are immunologically related treatments like anti-TNF, steroids, that sort of treatment. So if you treat the patients, their microbiomes return to normal. So again, it sounds more secondary than primary.
Would either one of you want to comment about what the future, the immediate future looks like in terms of sort of rendering the microbiome into a pharmacological entity? Are we on the verge of getting encapsulated, dehydrated human feces, and how are we going to standardize all of that? And
well, I think fecal transplants are. Oh, go ahead. Sorry. I'm not sure the future lies with fecal transplants. I've heard tell, and I don't really know this for sure, but fecal transplants have a certain amount of danger associated with them. And the FDA, wanted to rein them in a little bit, I think. So it should be done under controlled circumstances if it's done at all. But my guess is that this study with the Clostridia, has the ring of truth about it. And one of the things that we might see is the use of certain particular organisms to prevent disease. That would be very exciting.
So what I could add to that is actually now the field is also moving to try to create consortium of bacteria and to move away, because of course when we transfer the entire microbiome of someone, you transfer also things that you may not want to transfer, so viruses and elements that are undetermined. So now people are creating different kind of consortia of bacteria and trying to just test them as a group and to see if they can actually be used in a very controlled manner using, incubator that generate those in a very controlled manner. And that will happen. And this together, coupling one of the things that actually needs to be taken in account is to maintain a good microbiota. You need to pair that with the appropriate diet, because that's actually how you manipulate them. So I think having the right kind of microbe coupled with the right kind of diet I think this is something that's going to happen very soon.
Studies trying to correlate microbiome metabolome. There's a lot of people that are looking at metabolome as a way of determining what kind of microbiome they have, and try to correlate that to whether or not you're sick or healthy. I think that people are trying to make that.
Okay, well, thank you very much. This is very exciting.